Hey guys, so we're going to go over an environmental factors lab. Firstly, let's discuss obligate versus facultative. With obligate, it means that the bacteria can only operate under those conditions. So with aerobes, they can only operate in the presence of air. While, let's say, a facultative anaerobe would prefer to live in the absence of oxygen, it can also operate in the presence of oxygen. It just means that because it's facultative, that it would prefer the absence of oxygen, but in case there is oxygen, it can still survive. So as you would suspect, that it's easier to treat an obligate as opposed to a facultative, because when we change the conditions, such as when we use medications, the bacteria has another way to metabolize, while an obligate is restricted only to those factors. So for the next portions of the lab, it's going to be all the same. You're going to label and perform aseptic transfer. And with this one, you guys are going to have a lot of test tubes, so make sure to label them as far as what bacteria go in them and what, let's say that you guys are going to be using this for temperature, pH, and osmotic pressure. So you should label them in accordance to the temperature, the pH that they're going to go in, or the salinity concentration that they'll be in. So the aseptic transfer for this one's going to be a little different. You're still going to have your Bunsen burner, as you can see over here. You're going to still flame your loop. The difference in this one is that you're going to flame the test tubes. Both the test tube that has the microbes in them and the broth that you'll be inoculating them into. And so how that works is, let's say that this is your test tube. You guys will grab the cap off with your pinky, or your pinky and your ring finger. You'll pull it off. You'll already have flamed your loop. Take the loop, take the bacteria in, recap it, put it away with the other hand, get the other test tube, take it off, flame, inoculate. Take it out, recap, and then that's that. You'll be doing that pretty much for the whole thing. If it's a little difficult for you, try to work in pairs so that one can pass the test tubes over to the other person. If you guys have any questions about that, please let me know. But for the temperature, the pH, and the osmotic pressure portion of the lab, they're all going to be going in this order. So with the temperature one, you're going to be looking for growth at specific temperatures. If it's in the range of 0 to 30 degrees Celsius, you're dealing with a psychrophile. 15 to 45 degrees Celsius, a mesophile. And anything above 40 degrees Celsius is going to be considered a thermophile. In regards to the pH, anything below a 5 is an acidophile. In the 6 to 7 range is a neutrophile. Anything above 8 is an alkalophile. And in regards to osmotic pressure, anything that grows at roughly the 3% concentration is considered a halophile. Now there are extreme halophiles which are in the realm of that can survive in the realm of about 15%. And then we have osmophiles that can survive in a wide range of salinities. In the next part, you're going to be inoculating bacteria onto a plate. So same as before, you'll flame your loop, flame the test tube, and then you'll take the bacteria from the broth and inoculate it onto the plate. We're going to be doing this 3 times because we're going to have 3 plates that go into the torble jar, the candle jar, and the environment. The environment is just going to be one that's left in like a plate holder somewhere on the desk or something like that. This is the way that it would look. You'll have this little peace sign here to divide it into 3s, and you'll squiggle on the bacteria. With your torble jar, that will be your anaerobic condition. Your candle jar will be your microair, and environment would be your aerobes. So we're going to see on the next lab when you guys come back is which bacteria grew in all 3, which bacteria grew in only 1 condition. With that, we'll be able to determine which is an obligate aerobe, which is an obligate anaerobe, and which is a facultative anaerobe or aerobe. Now in the meantime, you guys can examine your Theioglycollate tubes. And the Theioglycollate tubes show the relationship with the microbes and the air. So we have our E. coli, A. faecalis, L. plantarum, and C. sporogenes. We can see that E. coli grows throughout the entire tube, meaning that it's a facultative anaerobe, meaning that it would prefer to grow in the absence of air, but it can also grow in the presence of air. So that's why it grows throughout the entire tube. We have A. faecalis, which is an obligate aerobe. That's why it only grows at the top. L. plantarum is something that we would consider a microaerobe or aerotolerant. So it doesn't necessarily like to grow in the presence of air, but it grows in a little bit of air. While we have C. sporogenes, that is an anaerobe, and that's why it grows all the way at the bottom. For your practical, you should be able to remember the names that go with the appropriate growth conditions. So throughout the tube, E. coli, A. faecalis at the top, L. plantarum kind of towards the top, and then C. sporogenes at the bottom. When I took this class, I had to remember the names for all these test tubes. Finally, we're going to go over the Kirby Bauer. Here we're going to label our plates. Just follow the instructions from your professor, and you'll perform the spread plate technique to get a nice lawn on the plate, followed by using the alcohol and flaming your tweezers, then using those tweezers to place the antibiotic discs on the lawn, and then you'll repeat this for the various other microbes. Now the way this should look is just like this. For right now, let's just ignore this red right here. And what we'll want to do is spread the discs, which are represented by the black dots, evenly on the plate. And the reason why we don't want to place them too close together is because we're trying to see if the antibiotic alone is effective against this bacteria. And the way you'll do that is you'll use a ruler to measure the zone of inhibition, which is denoted by the blue circle in the clearing. You'll have a chart that you'll reference in regards to the measurements that you use on the ruler to say whether it's effective or not. Just because it has a big clearing doesn't mean that it's effective. You have to refer to the chart for each antibiotic. Now, if we were to place them too close, as we see here in the red, is that we could get a synergistic effect where two antibiotics that are normally ineffective against a microbe are now working together and are able to fight off the microbe. And that's something that we'll look at on a later date. But for right now, we're trying to avoid that because you just want to see the individual effects of the antibiotics. Now, I hope this video helped. If there's anything else that you guys need, feel free to shoot me an email.